as in the form of scaffold, it will be called as single guided RNA. And when it may complex with Cas9 protein, then only your complete technology with the name of CRISPR-Cas9 will be active. And how will we do the all the things which has been given? I'm not going to read it this all. Only I have to make the clarity about these things. Now, this figure is I have tried to make clarity about the functioning of the CRISPR-Cas9 in action. Why this Cas9 is in action? How it acted inside the genome from where at where you wish to edit the genome of your host. This Cas9, it is a RNA guided in DNA endonucleases. Cas9 is the Cas means CRISPR associated. CRISPR associated 9, this protein, this the RNA guided DNA endonucleases. Endonuclease cut the double stranded DNA. And if you go for the molecular structure, then it will be it is of 160 kilodalton molecular weight, which will be composed of 1368 amino acid, which has to be transcribed by 4 kilobase pair of this genomic structure, genomic value. And this is the, you may see over here the palm. Perhaps in next slide I have made more clear about that. Here you may see the palm TGG. Here we have taken TGG. Then TGG is the palm in my example. Then just after this palm will this initiate the activities, will act as the motif for this transcription of this one, the cutting of the your DNA of genomic DNA, which has to be edited, and then your endonuclease will act on the basis of guide of the this single guided RNA, which is the complex which has been formulated by CRISPR RNA and transactive CRISPR RNA. You may see over here that palm is always a triplet, and it will act as the motif, and then a proto-activation motif. And now after 3 this nucleotides just after the palm, this Cas9, this seeding will take place. What is the seeding? I will be discussing in later slides. Seeding means just joining this single guided RNA with the genomic DNA. So where this notch has to be made for the cutting the DNA by Cas9. Here you may say very clearly that we have taken NGG, that is palm. Then cut site is just 3 nucleotides after the NCC. And these after 3 nucleotides, seeding will take place. What is seeding? Actually, in the case of CRISPR RNA, there are 2 evident parts of CRISPR RNA. One part is made up of 10 nucleotides, which is known as seed. And this seed will join on the basis of guidance of this sgRNA. And then after 3 nucleotides, this your spacer will lie, which is of 17 to 20 nucleotide, and at this place only this cutting. So that I'm saying again and again that it is very specific, site specific. There is no chance to go on the 4 nucleotide or 5th nucleotide, only first nucleotide. And if you are talking about the single guided RNA, in most of the cases it is of 100 nucleotide. Now the CRISPR-Cas9 system. What is this system? I told you that the PAM is your proto-spacer adjacent motif. These are the key organisms which decided the sites for the approaching the single guided RNA, whereas single guided RNA, they decide that this cutting has to be taken place. Yeah, cutting has the DNA has to be cut from where this message has to be taken by Cas9 protein. T9 I have told about. What is the forward engineering? In the case of backward engineering, you have to go for the recovery in the wild form, but you have to go for the mutation, for new mutation, for new alleles, for new genes, so that in the case of forward engineering, it is very much useful, in which singly you may go for the deletion. It means the cutting the genome and naturally joining the genome. Some part of in 1 or 2 nucleotide it may be cut, and the function of whole genome or whole gene, not genome, whole gene may be modified. And the modified form of particular event which is matching with your interest or with the value which has been decided by you has to be screened out later on. Otherwise, there is insertion of this nucleotide. If you are going to insert only 1 nucleotide, it means there is no going to any frame shift activity in the form of phenotype. Then you may go for the this simple without regulation. Whereas if you are going to insert complete gene, then there is requirement of the regulatory activities which has been decided by each and every territory and globe. As I've told you that I will take one example from for beyond this CRISPR-Cas technology, which is known as base. I have taken from the research paper which has been published by Dr. P.K. Gupta. This work has been done in 2019, very recently, in which they use a dead form of Cas9, a special form of Cas9, in which the cytidine deamination takes place, where this presence of cytosine is there, which will be converted in thymine. Then thymine will be paired. It will insist to homologous chromosome for the conversion as the adenine, and the vice versa in the case of DNA deamination, in the case of adenine thymine to this guanine cytosine. And this type of point mutation may create immense value of interest many times, which has to be taken by number of protocols and in various crops and organisms as well. Now, what is the assembly of CRISPR? We are talking again and again about the CRISPR. That we are taking the benefit of CRISPR the genome or CRISPR RNA or CRISPR gene, which is present in the this Streptococcus pyru, which is the pathogen of the human. They are pathogenic for the human being, but we are taking the advantage of the defense mechanism present or acting inside this bacterium. Then your CRISPR gene is present in about 40% of bacteria, which is using this technology is being used by 40% of bacteria for acquired immunity inside them from bacteriophages or various type of viruses. And in the case of 90% archaea, there is presence of, they are using this type of mechanism for immunity, for acquiring the immunity against the bacteriophages and various varieties of RNA, not only DNA, RNA, these viruses, but DNA viruses as well as the these RNA viruses. Then what is the structure of this CRISPR, this genome? The structure, as I have showed you, that in very initiation point, this is adjacent part of the CRISPR is Cas gene. Always Cas gene, your Cas gene is initiated when any bacteria or any virus or any bacteriophage come in contact with the bacterial cell, then your Cas become active and they give the information to CRISPR gene for its transcription. Then only is transcribed in the form of CRISPR RNA. If there is a leader and various repeats are there, I have told you the name has been taken from over here. Repeats are there, and these repeats are in palindromic forms. And between two repeats, there is presence of spacers. There are spacers, repeats, spacer, repeats, and any new viruses for which immunity has to be developed, these has to be incorporated in the genome of bacteria in the form of spacers. If there is presence of spacers, it means that particular virus from where spacer has been taken out, the bacteria will become immune against that particular virus. Okay, and now what is the process by which this immunity of bacteria has to be operated whenever it comes with the different pathogenic phages? I have given one example, arbitrary example, to make clarity about that. Just after coming in contact with the bacteriophages to bacterial cell, there is just ignition inside the Cas gene, and Cas gene, they started, they express the pre-cRNA. They insisted the precursor of CRISPR RNA, which has to be gone for the processing. After processing, it will become CRISPR RNA. Then this CRISPR RNA will go for binding with the initiation part of the DNA of the phages. And at that particular form, the spacer which has been present in the CRISPR RNA will be make a complex with the DNA of virus, and at particular sgRNA targeted points, your Cas9 will detach the DNA, it cut the DNA, and your bacteria will become immune and phages become idle outside the bacterial cell. And this is the very powerful technology, as we are using the antigen antibiotic interaction for our immunity. In the same way, the bacteria are they are using the this CRISPR-Cas complexes to create immunity among them for the bacteriophages, various type of bacteriophages. This is the technology which has been taken places synthetically for in various cases of crops and animal always. In the case of synthesizing the single guided RNA, for synthesis of single guided, we will talking about the natural process which are being taken place in the case of in bacteria. But here we are talking about the synthesis of the this sgRNA, how we are synthesizing. For this, for creation of, for operationalization of the single guided RNA, you will be requiring some operators, and it will be depend on your experiment and your protocols. Which type of operator will be requiring for a specific type of single guided RNAs. And for PAM, you have to decide that what type of gene you wish to target. It means number of activity which have to be operated in silico.